So let's get started. I will give you an overview of the past, the situation today, and a little bit looking into the future about cytotoxic drug exposure. But I have also decided to add another group of drugs, antibiotics, to my presentation. Because antibiotics is rather new when it comes to dangerous activities, exposure, and things like that in healthcare. And it's popping up the last 3 years, roughly, that antibiotics are maybe also, giving at first health effect when it comes to exposure. So I have put that into my presentation too. This is my background. I am not a nurse. I'm not a technician, pharmacy technician. I'm not a pharmacist. I'm a chemist. So I'm very interested in molecules and what they do and how you can produce them and how you can manage them. But I started in 1980 with my study chemistry, and at that time it was very usual that students were very laying back, so it took me 8 years to finish that. Nowadays it's not done, but that time it was a good time. Today it's maybe 4 or 5 years, but that time I was fine with that. So then I started my PhD. It was sponsored by the Dutch Inspectorate of Labor of the Dutch government, and it was the aim to develop test methods for validation of exposure to cytotoxic drugs. I finished that in 1993, and then later on, the university gave me the opportunity to make a business out of my PhD study, which I did, and I founded in 1995 Exposure Control. It's a company, a consultancy who was involved in monitoring exposure to cytotoxic drugs, and we do that nowadays around the globe. In 1996, I finally got my PhD. So this is a little bit about my background. The agenda for the cytodrugs today, there are many issues to address. Some will be a little bit more in detail, others will be more in an overview. Talk about toxicity guidelines, European Directive. I know European is a little bit controversial nowadays here in the UK, but anyhow, you're still in the European Union. So occupational exposure, sources of contamination. When you work with cytodrugs, where the contamination comes from, what is happening, where are the leakages, where are the problems, things like that. How do you protect yourself? Monitoring. Monitoring is actually, when you work with the drugs, you can monitor if you are exposed, you can monitor if the drugs are in the environment, so are there many options to do. I will focus much on surface contamination, a little bit about the references, what does it mean when you have these data. Also talk about exposure, real exposure of workers by urine testing. So if you do a urine test, you can see if people are really exposed. That's available nowadays. Closed system drug transfer devices, very hot issue nowadays. Cleaning, and I will end it with some conclusions, recommendations. That's the first part, and then the antibiotics will come. I have a question for you. This is the first one. So let's try if it's working. Which of the following statements are correct? Most chemotherapy drugs are carcinogenic. Most of the chemotherapy drugs are reproductive toxicity. Most of the chemotherapy have both, actually. There's 1 and 2. Most chemotherapy drugs are hazardous drugs. Or 5, all hazardous drugs are chemotherapy drugs. So it's up to you. Please press 1 till 5 what you think is the right answer. Some to go. I have only 50 votes now. So these are the results. So you have voted 44%. Most of the chemotherapy drugs are carcinogenic and reproductive toxicity, and 4 is the 53% said most of the chemotherapy drugs are hazardous drugs. The correct answer is 4. Most chemotherapy drugs are hazardous drugs. That's the right answer. Actually, 3 is quite okay too, but it's a little bit more than that. Here is the toxicity of the drugs, as effectively shown in a previous slide where you answered on. We have to deal with 2 effects when it comes to cytotoxic drugs. We have acute effects. Acute effects are effects that actually are found in patients. When patients are treated with cytodrugs, you get all these kind of effects, and sometimes it can be very severe, sometimes it can be quite okay. That's on the left column, acute effects. It's high dose, caused by high doses given to patients. The right column, the delayed effects, that are effects that happen after probably 5 years or 10 years or 20 years. That are effects you don't really see that much in patients. It happens there, but not that much. But it's more effects that are related to occupational exposure. So for pharmacy technicians, nurses who are working with cytodrugs day by day, by day, by day, after a certain period, there is an option that one of these effects happen. Reproductive effects, that is seen. There are studies published about it. Spontaneous abortions happen. Low birth rate is often seen. Prolonged time to pregnancy is often found. So it's really related to exposure for nurses, technicians who work with these drugs. And the other thing is carcinogenicity. It's very strange, but when patients are treated for cancer, sometimes secondary cancer appear after 5 or 10 years. And therefore, the idea is behind this that carcinogenicity is not really linked to only the exposure, high exposure levels in patients. But if you are exposed day by day by very small amounts, then there can be a risk for workers too. And actually, when you go to the classification, there is a classification on carcinogenicity, then you will find this classification by the International Agency of Research on Cancer in France. It's a part of the United Nations Association. And there it's indicated that all type of chemicals, but also cytotoxic drugs, can be divided in several classes. And class 1 is actually the class with the most dangerous drugs, because from these drugs or these compounds, it's known that they can really cause cancer. Then we have class 2, A and B, and these are drugs and chemicals that have been shown in animal studies that they can cause cancer in animals, and they cause cancer in animals, and therefore are probably also causing cancer in human beings. That's 2A, 2B. Then you have class 3 and class 4. So if we go, for instance, to class 1, we know this is just a few examples that these, let's see, 7 drugs are known carcinogens. So they're really the class 1, the most dangerous one. And I think when you take cyclophosphamide, etoposide, everybody knows that they are used frequently nowadays in hospitals. The others probably not that much, but cyclophosphamide, etoposide are really frequently used. When we go to class 2A, here are 4 types. I think cisplatin, doxorubicin are frequently used today now in hospitals too. So these are also carcinogens. Then we have class 2B. This is another example. Mitomycin, well known for bladder installation, is class 2B, carcinogenic to humans. Daunomycin, dacarbazine are not that frequently used. But anyhow, showing that it's a large group of drugs who are carcinogenic to humans and therefore also can cause cancer in people who are exposed to very low amounts day by day by day after 10, 5, 20 years. This is a very relevant sheet. Actually, it's talking about a specific group of carcinogens, so chemicals and drugs that can cause cancer. And it's especially the group class 1. The class 1 group, from these chemicals and drugs, it is known that when you get these in your body, that a very, very small amount can have an induction on your body to cause cancer. And for these drugs, it's very relevant to protect yourself in a very good way. And actually, from this information, it is decided that it's not only for these drugs, but it's for the total group of cytodrugs that you have to have that protection. which means that you have always to avoid exposure. So never get the drugs in your body or on your skin or whatever. You have to be well protected, therefore you take all these precautions nowadays. We have the guidelines, we have protective measures, we are working in isolators, we have cabinets and all these things. And in the end, there is also an obligation that if you work with these drugs and there are methods available, then you have to really check the workers if they are exposed or not. So, in fact, if you work with these drugs, like cyclophosphamide, which is one of the most, I would say, toxic one, but also cisplatin is also included in this group. If you work with these, then you have to be very, very much on the safe side. So that's the situation when it talks about a very specific group of carcinogens. Safety levels are described in the European Union Directive on carcinogenic compounds as 1. Obliged in a random order. 2. Obliged in a hierarchic order. 3. Advised in a random order. 4. Advised in a hierarchic order. Or 5. They are not present. Please give your answers. This is a tricky question. I have 50 votes now, I'm missing some. Well, 54, 55, it's coming. Okay. Good, okay, we will see. Oh, this is interesting. Quite a lot of opinions. Let's start with the majority. The majority said 42% advised in a hierarchic order. And the second group said obliged in a hierarchic order. So actually, the total group is saying there is some hierarchy in how to handle this, which is in fact true. The only thing is that every directive coming from Europe is not seen as a law. So that means that every member state within Europe has to make its own law and rules on a directive. And that means actually that the directive is not obliged, but it's advised in this case. So actually answer 4 is the right one. But it would be good that answer 2 would be the future, because that will improve your safety. And now I'm fine with these questions. So quite good. So when we go to that directive, which is already from 1990, so it's long ago, and there has been some updates nowadays. But anyhow, we go back to the past 1990. There you see what the strategy is within that directive. And it's about carcinogenic compounds, but as you know, most of the cytodrugs you work with today are also carcinogenic, so it applies for these drugs too. So the strategy is very simple. First, if you work with carcinogenic compounds, then you have to replace it by another one if it's possible. If it's not possible, then you go to the next step, and you have to reduce the exposure. So sources of exposure. So you make barriers between the drugs or the compounds and yourself, so you're not getting in contact with it. If you cannot do that, then you have to work on ventilation. So if drugs are in a room or whatever and you create enough ventilation, the drugs or compounds will appear. So they will disappear, sorry. They will disappear and get away from you. And finally, if all these things are not possible, you put on your personal protective equipment, your gloves, your gowning, all the things together. But that's actually the last step in the total strategy. If we apply this on carcinogenic compound, but then for the cytodrugs, so in practice for you who work with the drugs, then the replacement is not possible, because when a patient needs cyclophosphamide, you cannot say, well, this is not good for my safety. You get 5-FU because it's better for me. But that's not done, of course. So it's not really an option to change the drugs. So step 1 is impossible in oncology. So then you have to automatically go to step 2. And that means that you have to make the barriers between you and the drug. And nowadays there is only one option to do that, and that's working with closed systems. Closed systems is the only option that you make a barrier between you as a worker and the drug to get in contact with the drug. So it's a real barrier. It's closed behind you and the drug. Ventilation, that's what we've seen in many hospitals nowadays, is that a lot of hospitals work in room. You have clean room facilities, you work in isolators, you have cabinets, you have in the nursing areas ventilating the rooms and things like that. So ventilation is another option to get away of the drugs. But of course, there are many settings where there is no ventilation. And I will come later on that when it's about antibiotics. If you have no ventilation, but you have spill and leakage in your room, and you don't clean it, you don't remove it, then it starts to evaporate. So particles are getting around, you inhale it, or you get in contact with it. So if you have a bad ventilating room, for instance in nursing departments, which often is in very old buildings where you have no air conditioning or whatever, then it can be that contamination of drugs is increasing if you don't take it away. And in the end, what is done very often nowadays, people have a lot of things on their body, like the gloves, the gowns, mask, clothing, shoes, and whatever you can think about. But actually, this is the lowest way of protecting yourself. You have to do it, but you have to go higher up in the hierarchy. Actually, the first step now is closed systems, then ventilation, and finally, you get the personal protection. So when we look in Europe, and I think it's probably the same here in the UK, level 3 are mostly applied in Europe, and some hospitals apply level 2. So it's a really, really step to go forward to have implementation of closed handling. Okay, that was the directive, 1990, long time ago. It was the time that I started with my first study, so it's already a long time ago. But the directive will be upgraded and updated over the years, and they are working on it now to have also the mutagenic and reproductive drugs in that directive. So it will be a very comprehensive directive in the future.